Hello, everyone, and welcome to the online spinal cord injury research seminars. My name is Bethany Candelas. I'll be the person behind the curtain today. So today we're going to be having, one moment while I put up the slides. We'll bring on Amol Yadav is going to be our moderator today. Then put up our title screen. Apologize for the slight technical difficulty. Not quite as streamlined as Warren. So there we go. I'll let Amol Yadav go ahead and introduce our talk for today. Take it away.
Thank you, Bethany. Hello, everyone. I'm your moderator today, and welcome to yet another exciting session of the iOSCAR seminar. So today's speaker is Dr. David Magnuson. Dr. Magnuson is a professor and Franz Carmichael Chair in the Department of Neurological Surgery at the University of Louisville. He went to graduate school at the University of British Columbia, where he obtained his PhD in neuroscience, followed by first a postdoc at the University College London, and then a second postdoc at University of Ottawa in Ontario. Later, he started as assistant professor at the University of Manitoba before moving to Louisville, where he leads a research lab in the Kentucky Spinal Cord Injury Research Center. His lab focuses on studying neurons and pathways in the spinal cord that are responsible for locomotion, and then applying that knowledge of spinal cord circuitry and central pattern generators to understanding spinal cord injury and repair, and also investigating how activity-based rehabilitation affects locomotion function. So today he's going to share his research on a very exciting topic where he will explain the impact of physical therapy on locomotion after spinal cord injury. If you have any questions, please write them in the chat. Precede your questions with a capital Q or direct them to me with the @ sign. So without any further delay, I'm going to hand it over to Dr. Magnuson and can't wait to hear his talk.
Fantastic. I hope everybody can hear me well. Thank you, Bethany, and thank you, Amol. I'm going to stop my camera and start the talk. The next thing I need to do is thank Warren and Megan and the whole crew for this fantastic endeavor, the iOSCARs. It came at a great time to help mobilize and keep the field together. And I'll acknowledge it, the funding, most of the research I'm going to show today was funded by the DoD and the Kentucky Spinal Cord and Head Injury Research Trust, with a little bit of the early stuff funded by the NIH. So this is the obligatory dog picture. This is our dog Hope. My wife and I happen to watch Korean dramas together, so we call her Hopa. That's Hope growing up with our daughter's dog, Gracie. They live in Chicago. They come to visit quite frequently, although not as frequently these days, of course. And really, to get through the COVID situation, I rely on my granddaughters. That is Jane and Minnie and Naomi. And even though they don't live in Louisville, we get to use FaceTime a lot. And they're really the most precious things to me. So I'm going to give you a little bit of an insight into the influences in my scientific life. Hopefully this might help the trainees a little bit. I did my PhD at UBC, like Amol said, under the auspices of Hugh McLennan on the left. And he was a monumental figure in synaptic pharmacology in the 60s and 70s. Made some really big contributions to our understanding of synaptic transmission and long-term potentiation. He was a guy that just wanted to know how things work. And he wasn't really worried about diseases or anything like that, but really just how things work. And he was a true gentleman. So with that start, I went to University College and did a postdoc, and with Tony Dickinson at the Department of Pharmacology, which was a great experience. But one of the most profound experiences I had was meeting this guy, Rick Freit. He was living in the same building as I was. He was doing a postdoc at University College Hospital, and he has a C5, C6 asymmetrical injury. And I got him to approve that I could tell you about his story. So for around 10 months or so, I drove him back and forth to work every morning, battling the London traffic, even though it was just a few blocks. And I got to see Rick's day-to-day successes and struggles firsthand, the battles he had with spasticity and autonomic dorsiflexia, and the things that he had to go through on a daily basis. And that really set my trajectory to take my interest in just how things work and to move into spinal cord injury. And as you all know, the spinal cord injury field is populated with fantastic people, even Kareem, who really encourages craziness. And that's Kareem in one of our kayaks down on the Cumberland River in Kentucky. So here's the Magnuson Lab is all about teamwork. Here's the teams we're going to talk about today. Krista was a PhD student that got the whole stretching project started. Anastasia did the lion's share of the work. She published several papers on the subject. She's now a postdoc with Adam Ferguson. Greg States and Greta and Morgan are all PhD students that are currently in the lab and have had some part, large or small, in the stretching study. We have great core facilities at KSHRC. I've developed a really nice group of clinical collaborators where we're hoping to learn more about some of these things. I need to also acknowledge Scott Whittemore, our director, and one of my major collaborators, Jeff Petruska, who's really our afferent guru. So in the Magnuson Lab, we look at the spinal cord as being one of the key parts of the nervous system, and it is the part of the nervous system for locomotor function and for a lot of other motor functions. And here's how we look at it. The higher CNS, just let me switch to a laser pointer. The higher CNS sends commands down to the pattern generating circuitry in the lumbar spinal cord, which generates locomotor output. And actually, I point this down to say locomotor output, but we believe also, and we have some evidence from one of our other sets of studies in the lab, that the lumbar circuitry is also driving the cervical circuitry. That pattern generation involves rhythm and pattern and gain. So gain is for the load and weight support. Pattern is for the right-left flexor extensor pattern of the activity. And rhythm is the actual underlying tempo or frequency of step cycles. And a lot of people wonder about studying locomotion in a quadruped when really what we're wanting to repair is a biped. But really, I think for many aspects, humans are really just quadrupeds that happen to walk bipedally. And I really think that the circuitry that we see in the rat is very, very similar in its fundamentals to what we see in human. So this locomotor output is generating a load of afferent input that's coming into the spinal cord. And that's from the muscle, and from the joints, from the skin, the joint angles and changes, the contact with the walking surface, the load through muscle spindles and Golgi tendon organs, the velocity of everything that's going on. That afferent input is going into this spinal circuitry here. So the spinal circuitry is getting information from the higher CNS, and it's getting a lot of information from the afferent input coming into the lumbar spinal cord and then also into the cervical spinal cord during quadrupedal locomotion or bipedal locomotion with arm swing, etc. So that locomotor output that is generated can have changes that occur to it as a result of this afferent input. And can be refinement and learning going on. And these changes can actually occur with different timescales. And some of them we would think about as plasticity. There can be changing of a movement within the time frame of the actual movement if the intended output is not what is actually being generated. And then there can be changes that occur over time. And that plasticity, I think, is incredibly important after injury. After injury, the output to the pattern generating circuitry is reduced or eliminated. Therefore, the locomotor output is reduced or absent. Basically, this pattern generating circuitry at that point is really listening only to the afferent input coming in from the muscles, joints, and the skin. It's getting incomplete or confused information from the higher CNS. And because after injury, especially clinically, patients are often immobile for weeks or months, the afferent input that's coming into the system is really just what we are providing to that system. And in my lab, we believe, and one of our main hypotheses is that immobility contributes significantly to the overall functional deficits. And acutely, immobility sets a poor trajectory for recovery. So this is one of the driving ideas in the lab, and we think about the homeostasis of the motor circuitry no longer getting input from the higher CNS and having to look only at what's coming in from the periphery. So in other words, we need to be careful about what's coming in from the periphery, because that is what the spinal circuitry is actually seeing after spinal cord injury. So one of the most promising things we have for after a spinal cord injury is rehabilitation. And activity-based rehabilitation is repetitive application of appropriate movement, and that is providing this afferent input into the spinal circuitry. It's generating some movement that we see in terms of the movement or EMG patterns. And I think the models that we use to study these things are incredibly important for how we interpret what we see. We believe that rats and mice have a distinct advantage over cats and humans. Rats and mice, because they are low slung, they're low to the ground, if they make an error during stepping, the consequences of that error is quite minimal. Their belly hits the ground, they keep on going. Cats, on the other hand, and humans, if they make an error, it's quite devastating. Generally, they're going to fall over, have to get back up and start all over again. And of course, humans, it's even more devastating than with cats. So we look at this as, we look at the rats as a model that has some built-in advantages. Normally, we use a thoracic contusion, therefore the forelimbs are intact, and that provides a built-in retraining mechanism. So many years ago, I had a graduate student in the lab. He was a master's student, actually, John Kersey. And he, along with a whole team in the lab, did this very interesting study where we took animals, we gave them T10 contusions, and we put them in 2 inches of water. And this was supposed to be the absolutely not going to fail. It's going to be the best training strategy ever. This part was NIH funded. And the first thing we noticed is that when we took one of these animals with a T10 injury, and even at 1 week post-injury, when we put them in 2 inches of water, they could step immediately. It didn't look to us like they needed to be retrained. And that got us thinking about the capacity of the pattern-generating circuitry and how the capacity of the pattern-generating circuitry may be maintained, whereas the capacity to generate weight may not be maintained. And then when we used the shallow water stepping strategy as training, we were able to see that the shallow water training helped to maintain the early ability of the animal to step in shallow water, but it didn't bring about any improvement in overground stepping. These were animals with quite severe injuries. They had less than 10% spared white matter. And even with that low amount of spared white matter, they all, whether they were trained in shallow water or not, recovered weight-supported stepping. So the hypothesis is they were training themselves in their cages by walking around at night. And if I had read the literature, I would have seen this from Kareem, my buddy, when he was in Martin Schwab's lab back in 2000. They published this paper with over hemisection injuries and treadmill training, and couldn't see any additional recovery for the treadmill trained animals compared to the untrained animals. And Martin Schwab went on to publish some other papers about this kind of an idea. And he showed that if you build a rat condo, this was a paper by Starkey from Martin Schwab's lab. If you build a rat condo and you challenge the animals to go up and down stairs and to do various tasks in their cages, you can actually bring about additional training. So this is the proposed self-training through in-cage activity. To test that hypothesis, to show that rats are actually retraining themselves in their cages, Krista, a graduate student back in my lab many years ago, along with my oldest son at the time, who actually was the engineer who designed the wheelchair, came up with this idea of using a wheelchair. And if we put animals in wheelchairs starting 4 days post-injury, overnights only, as you can see here, we can dramatically influence their profile of functional recovery. And these animals were not put in wheelchairs. And you can see that this is quite a mild injury. This is the BBB score, the Basso-Beatty-Bresnahan score, that is pretty much standard in the field. And these animals recovered dramatic function. However, after 8 weeks of being in a wheelchair, this function was dramatically reduced. In a pilot study, Kristen noticed that some of these animals developed contractures. And so she said to me, Dr. Magnuson, these animals are going to need to have physical therapy like patients get to help prevent contractures. So with the help of Darren Atkinson and several others at the Fraser Rehab right across the street, part of our center, we developed a stretching therapy. And with this stretching therapy, what we do is we take a rat and we burrito it. We wrap it in a towel so that its tail and hind limbs are sticking out the back of the towel. And then we use one hand to hold the animal on its back, and we use the other hand to manipulate the hind limbs. You can see here iliac crest, hip, knee, ankle, and toe. This is a gastrocnemius stretch. Iliac crest, hip, knee, ankle, toe. This is a tibialis anterior, and this is a gastrocnemius stretch. And basically, each major muscle group is stretched. And when we stretched these animals, we didn't change the profile of recovery for the animals in wheelchairs, but we reduced the overall recovery of the animals that were not in wheelchairs. And that drop in recovery persisted when they came out of the wheelchairs after 8 weeks. This was really shocking to us. We didn't understand really what was going on. And one of the scary things is it didn't change the stretching of the animals in the wheelchairs did not reduce the incidence of contractures. And about half of the limbs got contractures in these studies. So this is a really scary thing to us. We had no clue at that time, and we still don't really have a clue if it's clinically relevant, but we thought that we needed to pursue it. Here's a little bit more detail about the stretching therapy, 6 positions, each major muscle group. The stretching is done to achieve normal end range of motion in the limb is positioned so that we're stretching the muscle and not the joint. Each muscle group is stretched for 1 minute, and then it's repeated for a total of 24 minutes of stretching. And at the time, this is many years ago now, this was modeled after what would be done in the clinic if funds and manpower were available. And we know, please don't get me wrong, we know that clinical physical therapy is much more than just muscle stretching. But this is what we have been studying, and we are basically now calling it muscle stretch-based physical therapy. So why is stretching done clinically? Well, it does induce a drop in muscle tone acutely, and it is perceived to maintain the range of motion. And there is something called stretch-induced hypertrophy, muscle hypertrophy, so it can help maintain muscle health. However, the evidence for a lot of these things are based on stretching strategies that are not the same as what's used in the clinic, stretching that's applied for much longer periods of time. And Lisa Harvey down in Australia published a series of papers several years ago and those papers basically did not support the clinical use of stretching after spinal cord injury based on range of motion. And this is a critical thing to understand, based on range of motion as the primary outcome. So we started to, at this point, Krista left the lab and we started to head down the road of trying to figure out more about the stretching. And the first thing we did was we tackled a study with Anastasia and the entire team in the lab to determine if stretching influenced locomotor function acutely and chronically. And this was a really interesting study. We started stretching one group acutely and then maintained another group unstretched, and it would become the stretched group chronically. The first thing that we noticed is when the animals have much more severe injuries, it's harder to get the end range of motion. And here in the first 3 weeks, our rat physical therapists, our rat hind limb stretchers, were stretching two-tone, the increased muscle tone that they observed in the hind limbs, and not past the tone. And we had one of the Fraser rehab folks come over, and she felt the hind limb, and she said, oh, well, you've just got to stretch past that tone. So when we applied additional force and stretched past that tone, we started to see more significant drops in locomotor function. And this is a really critical thing because this speaks to the time course of the changes. Monday morning, Monday afternoon, we did BBB scores both before and after stretching. And Friday afternoon, after 4 more days of stretching. The animals recover over the weekend, Monday morning, Monday afternoon, Friday afternoon. And then over a period of a couple of weeks, they recover back so that they are not significantly different from baseline. So clearly force is important, and clearly during the acute period, stretching is negative to the locomotor function. I stated in a lab meeting that I thought that once the animals are truly chronic and they're really stable, that the stretching would not be negative to their locomotor function, and I was entirely and incompletely wrong. So these animals were the baseline, were the control animals for the acute group. Once we started at stretching at 10 weeks, Monday morning, Monday afternoon, Friday afternoon, recovery over the weekend, Monday morning, Monday afternoon, Friday afternoon, these animals all scored 0 on the BBB scale. These animals basically, which showed small recovery over the weekend, basically were worse than animals that would have a complete transection in my lab. When we stopped stretching, we saw recovery, but there was some maintained deficits looking at the kinematics of the stepping with their hind limbs. So this was a devastating observation. We made some EMG recordings using telemetry devices, and we found that during stretching of the right limb, we could record EMG responses in the left limb, and this is clonic-like responses, air stepping-like responses, and spasms. And then at the end of the study, we actually stretched the limb that was instrumented, and we could record similar responses in the limb actually being stretched. So clearly this influences circuitry bilaterally. And it's very interesting that clonus is very rarely reported for rats, and we were able to see clonus regularly when we were stretching these animals. This is a little bit of an indication of how much force is being generated, and I can tell you 2 things. Number one, the amount of force that we apply is not different than what's being applied for human if you simply scale based on body weight. And fun with physics for the extensors, the forces that we apply are equivalent to the rat standing and gently bouncing on 1 foot. So clearly these are not extensive forces. And I haven't said anything about muscles, and I won't. If you want to look at how we demonstrated that the stretching does not cause changes in the muscle, please see the papers by Krista Call and Anastasia Keller. So far, we know that stretching induces a temporary loss of locomotion, that it works acutely and chronically for mild and moderate injuries, that it's force dependent, that it influences both ipsilateral and contralateral limbs in terms of the response to spasticity of kicking, but we don't know the mechanism. And right now, I have to give a shout out to Zev Reimer from Rick. Now, I don't remember the new name, the Rehab Institute in Chicago. He was visiting our center and he saw our stretching data and he said, oh, it's C-fiber mediated. And so we went down the road of trying to figure out if it in fact is. And this is another nutty monumental study done by Anastasia with an entire team of other graduate students and bioengineering co-op students in the lab. And this was done in collaboration with the Petruska lab. Basically, we took a bunch of rat pups, gave them injections of capsaicin. And if you do that, when animals are 2 days of age, they grow up without TRPV1 positive C fibers. And then we let them mature. We took the females of that group. We gave them spinal cord injuries. We let them recover for 6 to 8 weeks, and then we stretched them. And we had a group that received saline injections as pups, and that's this group here in gray. And you can see that they showed the normal sawtooth drop in locomotor function. We stretched during this period and then also during this period. However, the animals in red here that had received capsaicin at 2 days of age and have a dramatically depleted population of TRPV1 positive nociceptive afferents, which normally signal thermal and chemical pain. When those fibers are depleted, these animals showed a much smaller drop in locomotor function. It's interesting that the animals without C fibers showed a relatively normal, fairly normal recovery after injury, maybe a little bit of a drop here. So as you can see here, we stretched and then sacked these animals, and this was so that we could look at CFOS in the spinal cord as a marker of new activity. And then we also did immunohistochemistry for CGRP. Here's CGRP in the dorsal horn at L2 for the capsaicin animals. You can see that it's dramatically reduced for the control animals that didn't get capsaicin and didn't get stretched. And then for the vehicle animals that didn't get capsaicin, and these of course got injured. And then the vehicle animals that got saline and got stretched, they had a much higher amount of CGRP in the dorsal horn. And when we looked for CFOS positive nuclei, the vehicle animals that got injury, had saline at 2 days of age, had a much larger number of CFOS positive nuclei, suggesting neural activity in the intermediate gray matter, and for that matter, even into the ventral horn down here. This is the central canal and dorsal horn. That capsaicin animals that showed a much smaller drop in locomotor function had fewer CFOS positive nuclei. So think about this. These animals basically aren't stepping because of the stretching, and they have a dramatic number of CFOS positive nuclei in the deep dorsal horn. So there's a bit of a summary slide, and we have come to the point of mid-seminar questions. I hope there are some questions. I don't hear anybody.
Yeah.
There we are.
Thank you, Dr. Magnuson, for the great start to your first half of the talk. And so, yeah, we have got a bunch of questions that came in during your talk. So I'm going to go through them one by one.
Okay.
And then you can answer them. So Kathleen Cole is asking, instead of manual stretching, did you ever try allowing the wheelchair-bound rats access to shallow water stepping?
That is a fantastic question, and it is a study that we have wanted to do for the past 10 years, and we have not done it. Once we moved away from wheelchair, we have not gone back, and it's a great idea. We are now using tiny cages to restrict the activity of animals, but that is not like a wheelchair, and it's not as clinically relevant as a wheelchair. So Kathleen, that's a great question. We have not done it.
Okay, so there's a question from Bharadwaj Nandakumar. He's asking, what are the parameters for mild injury? Is it 100 kilodyne?
Our mild injury for the rat was 12.5 gram centimeters. That was Krista's original paper, but I will tell you that we were using stabilizers that stabilized the spinal column that were not sufficient at the time. So the animal got a more mild injury than what they should have received. Now, they all received the same injury because we used the same stabilizers with all of them. So it was a 12.5 gram centimeter injury, but honestly, based on the spared white matter of Krista's paper, it was more like a 6.25 gram centimeter injury.
Okay. Can you switch on your camera? I think it's blank.
There we go.
Okay, great. So there's a question from Ona Bloom. In humans, there are interactions with muscle atrophy and bone loss. So did you see any changes in bone in the animals that didn't recover?
Great question, Ona, and I appreciate it. We did not look at bone. I have done a little bit of work with bone, and it's amazing how little activity the animals need to maintain normal bone. We will see 10 to 15% bone loss, but beyond that, we really have to go to a very, very severe injury. So no, we did not look at the bones. Now, we did look at the bones in the wheelchair study, and you would have to look back at that paper. I can't honestly recall. It's part of the supplemental data in Krista's paper.
Okay, there's a question from Juan Herrera and Hadi A. Similar questions. They're asking, did you check pain response in these animals because of the C fiber effect?
Yes, we did, and that's something that we're getting better at. We are, at the time of the studies that I've talked about so far, we were very rudimentary in the way we tested for pain and nociception, and we did not see differences. There were trends, but we did not see differences. We're getting better at it, and with my current group of graduate students, I believe that we will be able to demonstrate differences.
Okay, and then there was a question from Dana McTighe. I think it was in response to a comment from Morgan, and she's asking, do you think the tiny cages increase stress response?
Hi, Dana. Thanks for the question. I am, we're certain that tiny cages and the wheelchair would change the stress responses. We have not quantified that. And in a way, it's partly because we feel sort of like people are going to be under stress as well. We do controls so that we, for instance, I'm going to tell you in a few minutes about burrito controls so that we wrap the animals in the burrito as if they're getting stretched, but we don't stretch them. So we have tried to control for it a little bit, but there's no doubt that the stress is increased. And I'm sorry I don't have a better answer for you.
Okay, and then so next question is from Lynette Montgomery. And the question is, do you stretch both hind limbs? And what would be the effect on locomotion of just stretching one hind limb, as afferent inputs would be different between the stretched and non-stretched limb?
Hi, Lynette. Thanks for the great question. That is part of our current DoD grant to try to sort out if the stretching effect requires all of the muscles to be stretched. For instance, we're going to stretch hips versus ankles. We're going to see how that influences CFOS in the spinal cord. So right now, I'm afraid I can't tell you. All of the stretching we've done up till now has been all of the muscle groups on both limbs during each stretching session. So good thinking, though.
Okay, so for the sake of time, I'm going to ask one last question before we move on to the next part of your talk. So this question is from John Walker, and he's asking, how do you think the C fibers are mediating an effect on motor control? Like, what is the mechanism? And do you think the results with capsaicin are a result from development of adult C fiber activity?
This is a great question, and we don't have a lot of direct evidence for this kind of thing yet, but I believe that the stretching the effect of the stretching is dependent in part on the post-injury sprouting and hypersensitivity of the C fibers, but how that sprouting and hypersensitivity of the C fibers actually results in a decrease in locomotor output, we don't yet know. I think it probably has to do with the overactivation or aberrant activation of these interneurons that wouldn't normally be activated during a stretch, but because it's post-injury are, and that somehow renders them unable to continue to participate in locomotor function. That's just our hypothesis, just our guess.
Okay, that's great. I think so. We have a ton of more questions, but because we need to move on, I think you can come. You can come back later and answer them, or some of your students and trainees can respond to them in the chat. So let's move on to the next part of your talk.
Fantastic. Okay, so from here on, the work I'm going to tell you about is unpublished. And the first study was to evaluate if stretching has a similar impact on locomotor function following a T2 injury, which we consider to be more clinically relevant than a T10 injury. And this study was tackled by Morgan Sharp and Greta Caesars, two relatively new graduate students in my lab. And they did a great job of this study. As you can see here, we had a control group that remained unstretched, and we had a T2 that they both received T2 injuries. And you can see that we see this normal sawtooth pattern for the stretched animals: Monday morning, Monday afternoon, Friday afternoon, Monday morning, Monday afternoon, Friday afternoon. But the overall loss of locomotor function is not as great as we saw previously following T10 injuries. And these animals seem to recover fully over each weekend. So yes, it looks like, and this appears to remind you of what the chronic T10 animals looked like. So clearly this is not as severe a drop in locomotor function as we see here. And so yes, it's negative to animals with T2 injuries, but it doesn't seem to be as negative, and there seems to be a little bit more variability. And we thought, because we were using T2 injuries and that their locomotor function would be dramatically impaired, that we might see cardiac output changes in stroke volume, etc. So the monumental task of doing echocardiography on all of these animals showed no change in cardiac output or stroke volume or any of the primary cardiac indices that you can get from echocardiography. So the next study we undertook was to look and to try to determine if spontaneous activity interacted with the negative impact of stretching on locomotor function. And so over a number of years and a number of different bioengineering co-op students and graduate students, my lab developed this strategy of utilizing tiny cages and extra large cages as ways to manipulate the overnight activity of the animals, and we can estimate their activity here. These animals are always paired housed, and we use ultraviolet, no, I guess it's infrared cameras. Anyway, we use overnight cameras, and we spy on these animals, and we can know what their in-cage activity is overnight. And it turned out that if you put an animal in a tiny cage, yes, you're reducing their horizontal activity, but their vertical exploration increases. So we actually developed a tiny cage with a low ceiling. So in the study I'm going to tell you about, we've got two groups of animals, one housed in large cages and one with normal height ceiling, and one housed in tiny cages with low ceilings. And this gives you an idea of how we can change the activity. And again, this is post-injury, 4 weeks post-injury. These animals were getting around 200 meters a night, and animals in the tiny cages were down around 50 or 75 meters a night. So it's a substantial difference in overnight activity. And when we did that with these injured animals that are receiving stretching, we saw no difference in the negative impact of stretching on locomotor function for the tiny cage drop ceiling animals in red and the large cage with normal ceiling in black. And this really surprised me. I really expected that the additional afferent input, appropriate, good afferent input coming into those hind limbs while the animals are walking around in their cage at night would influence the impact of stretching. It's interesting to note over here, this is the distance traveled overnight. And I believe this was at the 8-week time. Oh no, this is over 5, 6, 8, and 10 weeks post-injury. When we first started stretching, the differences were quite significant. But at 8 weeks, the in-cage activity of the large cage animals actually decreased, so that the differences, even if it's statistically different, it's not as big a difference as we would like to see. So then we decided to tackle something to do with the pharmacology, and this is partly the influence of Jeff Petruska's lab, but of course he wouldn't have gotten behind this hypothesis at all. This is a harebrained hypothesis that the boss came up with. Sprouting of nociceptive afferents that are also hyperexcitable leads to overactivation of substantia gelatinosa interneurons and excess release of endogenous opioids, which in turn reduces locomotor output. So my idea here is simply that all of that sprouting and the excess activity of the nociceptive afferents would lead to the system being flooded with endogenous opioids, and that would cause a drop in locomotor function. I mean, you don't want to walk around when you've got opioids. Anyway, so we decided to test that with a broad spectrum opioid antagonist called naltrexone, which is very well known. It's been used for many, many years. And we were able to find some lovely published studies showing the IP dose. And so what we did was first of all, we burritoed these animals for a week to make sure that we weren't seeing any stress-induced drop in locomotor function. And then half of these animals in this small N pilot study got naltrexone injections before stretching, and half of them got saline injections, and we injected them half an hour to an hour before the stretching sessions. We did kinematics and, of course, BBB. And this study was run by Greg States, one of the relatively new graduate students in my lab, who also has an engineering background. So here's what we found. Here are the animals' baseline after injury. We saw the usual drop at 1 week post-injury. They both recovered. These animals should all be the same because there was no difference yet. Then they started to get burritoed on a daily basis as if we were stretching them, and we saw no change in locomotor function. Then here we started to inject saline for the animals that you see in red, N equals 4, and naltrexone for red, N equals 4, and saline for black, N equals 4. And they both showed the expected drop in response to stretching, but it does appear that the naltrexone was a little more robust. It's not statistically significant. We let them recover so we could see that recovery profile. And then on that first day of stretching in week 8, we saw a larger drop in locomotor function for the animals that were receiving naltrexone. Both groups showed the expected drop, but as you can see, the maximum drop showed by the naltrexone animals and the profile of recovery. And then again, when we started stretching at week 11 so that we could sack these animals to look for CFAs, we saw that the naltrexone animals showed a more robust negative response to stretching. So Greg did some nice immunohistochemistry. These are the saline animals and the naltrexone animals, looking at the immunohistochemistry for CGRP. And believe it or not, it looks the same. And when he quantified it, showing the heat maps here, and when he quantified it, in fact, there was no difference in the CGRP in the dorsal horn, which really surprised me. Keep in mind, these animals all got injured. They all got stretched. The animals that had a larger response to the stretching at some time points did not show increased CGRP for the animals that got naltrexone. We then did some beautiful immunohistochemistry for CFAs, and these are the nuclei with hooks and CFAs in green. And in this case, the naltrexone animals had a much larger response and many more CFAs positive nuclei down in the intermediate gray matter. And there's the heat map that we showed for the iOSCAR slide. These are CFAs positive nuclei from animals that are being stretched after injury that received saline or that received naltrexone 30 to 60 minutes before the stretching session. And Greg did some beautiful little heat maps, contour plots, and this is to help us understand if there was an expansion of the region of gray matter where these CFAs positive nuclei were showing up, or if it was really just a higher concentration. And it looks like it's a higher concentration. There might be a little bit of an expansion for the naltrexone animals down into the ventral horn. I don't believe he's done statistics on these yet. So there's the CFAs counts. When I say statistics, I mean versus the regions dorsal ventral. These are the CFAs counts for L2, L3, L4, L5, and the overall total. And you can see that the animals that got naltrexone had a higher number of CFAs positive nuclei in the intermediate gray matter compared to the animals that got saline just before being stretched. So the results, clearly the harebrained hypothesis was clearly wrong. The locomotor drop appears to be enhanced for the animals that received naltrexone. It was at a couple of time points, but not at every time point. We saw no change in CGRP, and we saw an increase in CFAs positive nuclei. So these results suggest that endogenous opioids may actually be helping to maintain locomotor function after spinal cord injury and stretching. And as Jeff would probably argue, that's logical since opioids are anti-nociceptive. And we assume at this point that the stretching is having a negative impact via activation of the nociceptors. It also suggests that there may be a disconnect between CGRP afferents, their sprouting, and the endogenous opioid mechanisms. So, of course, something on the list of experiments to do is to actually give the animals opioids before we stretch, and that's something that that Morgan has planned for her future. So, in summary, let me get my summary page. In summary, stretching influences locomotion negatively in mild to severe injuries, in acute and chronic injuries. After T10 and T2 injuries, the responses, the drop in locomotion are temporary, they are force dependent, they are dependent on TRPV1 positive C fibers, they are not prevented by high levels of in-cage spontaneous activity, they may be related to increased CGRP in the dorsal horn, they certainly seem to be related to increased CFOS positive nuclei in the intermediate gray matter, and they may be exacerbated when opioid receptors are blocked. So clearly, stretching in rats, hind limb muscle stretching in rats after an incomplete spinal cord injury is bad and ugly. The question is, any good? And the one good thing that we can point to comes from Anastasia's paper back in 2017. This is the acute and chronic stretch paper. And she showed that using something that we called the magnetic tail stimulation response, it's where we magnetically stimulate the base of the tail and we look at EMG responses in the hind limb muscles. The animals that were receiving chronic stretch showed a drop in EMG responses in response to magnetic stimulation of the base of the tail. That suggests that the excitability of the motor system below the injury is influenced by stretching, and that would speak to the potential for stretching for reducing spasticity. So in the rat, physical therapy focused on muscle stretch after an incomplete injury is bad and ugly. And we hope that this work and that of the work of Jim Grau, Mike Beattie, Adam Ferguson, Megan Detloff, and many others, we're hoping that this will prompt clinical studies into the role of the afferent input, acute activity and inactivity, and what we do for physical therapy and with straight patients in the acute period. So hopefully we can alter that trajectory of recovery during that acute period and improve the balance between adaptive and maladaptive plasticity. So thanks everybody for hanging in there. And in particular, thanks to the really great trainees that I have had and have in the lab. They challenge me every day, and they challenge me and each other every day to be better scientists and better people, and have really contributed to the field. So with that, I'm going to skip through to another photograph. That is a photograph of Point Reyes National Park in California. And I would be very happy to answer any additional questions.
Okay, great talk. So yeah, we have a ton of a lot of more questions. So I'll go through them one by one, and you can answer them. So there was a question before the break, before starting the second phase. So I want to get to that first. So the question was from Guillaume Caron. Did you try to inject in rat metabolite to activate muscle afferents type 4 during the stretch? That is lactic acid or KCl or bradykinin?
That is an absolute awesome question. We really do need to and want to get at what's going on in the periphery and what's going on centrally. And clearly that idea of using metabolites or using, capsaicin or something to activate the muscle afferents in the periphery is a great idea. And it's just something that we haven't done. If anybody wants to do it, let's talk, do it. But great question, Guillaume.
Okay, so there was a question from Lawrence Moon. And the question is similar to a previous question about CFOS, the question is, what the C-FOS pattern was very widespread. So are C fibers suppressing premotor neurons?
Dr. Moon, good to hear from you, and thanks for the question. Our hypothesis is that the activation of the interneurons, either directly by the C fibers or indirectly via other interneurons, spreading down through the gray matter into the intermediate gray matter, is somehow inducing those interneurons to be less responsive to the desires of the pattern generating circuitry, as either as part of this pattern generating circuitry itself or as intermediators between the pattern generating circuitry and the motor neurons, so that they can no longer respond normally to the pattern generator output. That may involve things like chloride homeostasis. That's one of the main things we think about. Maybe changes to 5-HT. There are many other things that that we are looking at, and we'd love to have collaborators to help us sort these kinds of things out. So great question.
Okay, next question is from Megan Detloff. She asks, how does not having C fibers during development affect pain and sensory motor function?
Well, we used, that's a great question, Megan, thank you. We used what's called the cutaneous trunci muscle reflex that Jeff Petruska has in his lab to demonstrate that these animals are unresponsive to thermal pain. And that's thermal pain delivered to the skin. And this is in otherwise intact animals that received capsaicin at 2 days of age, but did not yet have a spinal cord injury. So clearly these animals do not respond to thermal pain. So their heart gaze response, their tail flick response, all of those things are basically gone. They still respond to mechanical pain cutaneously. And it's in those animals that when we stretch, the drop in locomotor function is dramatically reduced. And it's a very interesting thing when the animals, before injury, when these animals got to maturity, the males all started to die because they couldn't pee. And it seems like the TRPV1 positive C fibers are necessary for male rats to pee, even without spinal cord injury. And then with the females, after spinal cord injury, the TRPV1 positive depleted animals had to have their bladders expressed every day for the rest of the study. So they couldn't develop a spastic bladder without TRPV1 positive C fibers. So it's a very, very interesting model, and I would love to see other labs adopt it to answer some of these questions.
Okay, great. Next question from Guillaume Caron. Did you test whether stretching decreases muscle passive resistance?
That is another great question, Guillaume. And I believe it is in Anastasia's acute chronic study, she demonstrated that the end range of motion that could be achieved for animals that received the chronic stretching was reduced. So the range of motion of the limb was reduced following those weeks of stretching compared to the animals that were not stretched. When we stretched, the only muscle group that we saw increase in force were the quadriceps. Otherwise, the amount of force we were applying didn't seem to need to increase to achieve end range of motion, but the end range of motion that was achievable did decrease over time. So it's very counterintuitive from what, one of the arguments for using stretching clinically, which is to maintain end range of motion. Great question.
Okay, there is a question from Ronaldo Ichiyama. And the question is, were there changes to muscle spindle structure, function, output, and input to motor neurons when stretching interventions were administered?
Ronaldo, great to hear from you. Thank you for the question. It's not really a very good question, but thank you for the question anyway. We did not look at muscle spindle function or the muscle spindle afferent output. The only thing, what we have done is look for muscle damage. So we've looked for fibrosis in the muscle, we've looked for inflammatory infiltrates into the muscle, and we've looked for centralized nuclei in muscles that have, many weeks after stretching, and we have not seen any indication that our stretching paradigm is in any way damaging the muscles. Now, that doesn't mean that muscle spindle output is maintained. It may not be. But we had to make a choice, and after Zev Reimer said, oh, it's the C fibers, and I had some other people that will remain nameless indicate they thought it was muscle spindles. Well, we chose to go down the C fiber avenue and haven't looked back since. But it is a good question. The muscle spindle function may be altered. We don't know.
Okay, and then this question from Megan: what does anything happen to the rat's weight when they are in tiny versus large cages?
That is a great question. You would assume that there are weight changes, but I believe that they are insignificant, that they're non-statistically significant. We would need to find out from the trainees. I don't have that information on the top of my head. I apologize for that.
And then this question from Hadi, and the question is: did you check for gender differences? Because in opioids, there's this issue with different response.
And we have not done that. The Magnuson lab has been recalcitrant to include males, except for really key control groups in some experiments. We've used some males with some of our T2 injuries to look at the cardiovascular responses versus females. None of the stretching studies have involved male animals. Now that we know, or we suspect, that the opioids are involved, I believe that this is something that needs to be done. I agree with that.
And so a follow-up to that, what do you think is the source of the endogenous opioids?
Well, after even before injury, the substantia gelatinosa has these beautiful, tiny little interneurons that generate and produce endogenous opioids. And I'm assuming that it is those interneurons. You know, one of the things that you carry with you are your experiences as a postdoc and a graduate student. And I learned so much in Tony Dickinson's lab, and working, interacting with people like Cliff Wolf and Steve Hunt. And I mean, it was just such a rich environment. And so we got to know those interneurons quite intimately. And I'm just assuming that those interneurons are releasing endogenous opioids into the system, and possibly as a result of the C fiber activation during stretching. So if we add exogenous opioid, we may actually be able to reduce the negative impact of stretching. We're just going to have to continue to investigate that avenue.
Okay, and then there's Bharadwaj Nandakumar is asking, is there a general consensus on the role of physical therapy to the recovery of function? Is treadmill training insufficient, or do you have any comments on the duration of training and the time of the injury?
Well, that is a very loaded question. I am not going to comment on the treadmill training. I'm a real proponent for do things early, get the animal, get the person moving early. One of my clinical collaborators up in Montreal, Jean-Marc Macthiong, is trying to use cycling very, very early post-injury. And I think this is a good thing to do, to try to get good afferent patterns into the spinal cord acutely. I don't think there is a general consensus for the role of physical therapy after injury. And one of the things I've learned, I've been fortunate enough to be invited to talk at some largely clinical meetings that are populated by both physical therapists, physiatrists, and rehabilitation physicians. And I think the approach that many centers take is dependent on that center and the leader of that center, and where the people that are in that center or that hospital were trained, and how much interaction they have with people that are familiar with basic science literature, et cetera, et cetera. I think we don't yet know if this is clinically relevant. And we really sorely need to try to figure that out. I think it is using fundamental circuitry that probably is relevant for both rats and people, but we need to definitely figure that out. So I don't think there's consensus. And in terms of rehabilitation, I just say the earlier the better. So I'm sorry I can't give you more than that.
Okay, so we have reached 3 p.m., but I do want to make sure I get one question that's from my perspective a little bit. I as a neural engineer and coming from the area of brain-machine interfaces. So what do you think is the the role of sensory feedback in improving locomotion after injury? So what if we from create a brain-machine interface where there's feedback directly going back to the brain about the injury?
So I have real mixed feelings about BMI work and the role of engineering if it excludes or somehow doesn't involve fully the locomotor circuitry and the lumbar enlargement. The afferent input coming in from the muscles and skin is critical both for what's going on in the brain, or as I like to say, up in the coconut, and down in the lumbar circuitry. Bypassing the locomotor circuitry, I think, is a shame. I totally understand that for some patients that may be necessary in order to bring back certain aspects of function. But I really hope that we can work with BMI and the locomotor circuitry, with the coconut and the lumbar circuitry and the afferent input together to improve things. And I think what's going on now with epidural stimulation and transcutaneous stimulation is just the tip of the iceberg of what's going to be possible. The work of Vivian Mushawar and other people that have done interspinal stimulation as well. I think we're going to be able to bring all these things together. But my hope is that by addressing things acutely, the trajectory of recovery for many of these patients will already be improved, and therefore the additional improvement that is necessary will actually be less. So that's one of my main things. And I need to apologize to Jeff Petruska because I'm speaking out of school, and he's already offered to do some of the things that I've said I'm looking for collaborators for. So anyway, that's a good question, Amol. Thank you for it.
Okay, great. I think we have reached the end of the talk. And I do want to say that, you can stick around in the chat and answer some questions that may not have been, we didn't have time to get them through in the chat later. And so next week, we have a talk from Dr. Shelly Sakiyama Albert from the University of Texas at Austin. And so, we are really looking forward to the talk next week. And then I'm handing it over to Bethany.
Okay, thank you so much. That was a fantastic talk. I always love when it makes my job hard because the talk is so interesting that it's hard to keep track of all the questions and everything. I did just want to call attention to, if you missed the announcements at the beginning, that you can sign up for email alerts. If you're already receiving emails from us, you still need to refill out this form. I saw that some people have been dropping it in the chat. We will be transitioning over purely to email addresses that we received from the form and not from our previous setup. And then I also saw that we had the links for nominating a speaker, so please do that as well. And you can also get all these updates both from getting email alerts and also following us on Twitter. And with that, I'd like to say thank you again to an absolutely fantastic session, and we will see you all next week.
Thanks, everybody.
Thank you. Bye.
Thanks, Amol. Well done, man.